Welcome to the In-Depth Academy. Today, I'm absolutely delighted to be joined by Francesca Lopez, who joins us from the National University of Ireland in Galway. And she's going to be talking about her research for a short while. And Francesca is a previous recipient of a STSM, so a short-term scientific mission funded by the In-Depth Cost Action, which of course is what the In-Depth Academy is based upon. So, as I said, Francesca is going to give an introduction to her research, as you can see on the screen, and then I'm going to come back in a little later and ask her a couple of questions about the STSM. So thanks so much, Francesca, for joining us, and I will
Thank you.
Later.
Thank you, Geraint. Thank you for giving me this opportunity. So my name is Francesca. I'm a PhD candidate at the National University of Ireland in Galway under the supervision of Professor Charles Spillane. And I will talk to you a little bit about my work with Arabidopsis thaliana populations with feature reduction of 45S copy number the ribosomal genes. So just to give you a quick introduction on my work, the ribosome biogenesis, the process of ribosome biogenesis, begins with the transcription of 45S precursor ribosomal RNA. This is transcribed from the gene called 45S rDNA, which comprises the subunit 18S, 5.8, and 25S. And these genes are very special because they are one of the most abundant repetitive elements in the genome. And there's a lot of evidence emerging that they're not just there to make ribosomes, but they actually have an important function in the architectural and structural organization of the genome. So in plants, these are quite well characterized, and they are quite an old topic of research. But it's in Arabidopsis, we have very large differences in copy number across different accessions. In the accession called 0, the genes are arrayed on the short arms of chromosome 2 and chromosome 4, with an interesting developmental regulation of transcription. So because these genes are not all of them are required for active transcription, a large number of them become silenced through heterochromatic modifications, and especially and DNA methylation. And so in col 0, this phenomenon occurs about 20 days after germination, and all of the genes on the chromosome 2, so called NOR2, they become silenced. So at the beginning of my research, we hypothesized that by using CRISPR-Cas, we would be able to modify the copy number of ribosomal genes. Specifically, we used a promoter that is specific and only active in the egg cell, so inside the ovule, and we use a single guide RNA that is specific to the 18S region. And so we hypothesized that through sequence identity, we would be able to generate extensive random double-strand breaks across the repeats, generating large deletions of copies of genes. And simultaneously, through the action of non-homologous enjoining, we hypothesized we could recover plants with supernumerary copy number. And indeed, in the T1 population, we retrieved plants with very large variation in copy number, from 20% up to 160% of wild type. And so the first thing that we did, we wanted to see if we could reach a plateau in copy number. And so we selfed these plants for several generations, and we retrieved a T6 population with a plateauing copy number around 10 to 15%. And so one of the first analysis I was keen on carrying out is a cytogenetic analysis, especially to investigate that developmental regulation of transcription. And the first step I took towards this was to sign up for the in-depth 3D fish training school in Clermont. That took place in 2018. And because it was a technique that I had never used before, that was really instrumental for me to learn how to do fish. And through that, we also started a collaboration with the lab of Aline Probst and Christophe Tatou. And through the short-term scientific mission, I was able to generate really important data for my research, especially these beautiful fish images that we took in Clermont, which show that this developmental regulation seems to be altered in our lines. And you can see that all the signals, the rDNA signal, shows at the nucleolus, which suggests that all the copies are available, at least for transcription, if not actively transcribed. And in my second STSM, we were interested in exploring the epigenetic landscape of the repeats, and we actually found that there is a drastic loss in silencing mark, such as K9MA2, which is a silencing histone mark. And so tied with, with this, it kind of gives us an explanation of the fact that there is a maintenance of gene dosage of ribosomal RNA. So effectively the production and accumulation of ribosomal RNA is the same as wild type, and most likely it's through changes in epigenetic landscape that we are able to maintain this ribosomal synthesis. Further, with also through collaboration with INDEPTH, with who I consider to be the most authoritative figure in the field is Clermont-Ferrand, of course. We were able to sequence both lines, two lines that we selected, line 236 and 289. We were looking really for signs of genome instability. We actually retrieved in one of the two lines a large segment, chromosomal segment duplication on chromosome 4, and this only appeared in the T5 generation, which is telling us that it could be a sign of genomic instability. And this is the line of research that we're currently investigating, and it's quite interesting. And so I could have shown you a lot more, but I'm going to leave a bit of suspense. You might be able to read it soon enough, and I would definitely like to thank you, Garrett, for giving me this opportunity, and especially thank my supervisor, Charlie, and all my colleagues at the lab, our NUIG collaborators, Antoine, Marcus, and Ronan, our, of course, INDEPTH collaborators, Aline and Fred, and also collaborators in Milan, Luca and Paolo. So thank you very much.
Great, thanks very much, Francesca. You truly are a poster child for the in-depth cost action. So as you mentioned in your talk, you did a couple of STSMs. Can you let us know? Can you restate where you did those and what you did in those STSMs?
Sure. So I did both my STSM in Clermont-Ferrand in the lab of Aline Probst and Christophe Tatou, and my second STSM was more of a really continuation of the work that we started during the first one. They were both really instrumental in generating really important data for my research. Yeah.
Okay.
That was
So, I mean, I think a lot of people will do an STSM, and then maybe that will be the end of the relationship with the lab they went to. But have you had an ongoing relationship with Aline and Christophe?
Absolutely. Yeah. We really, it was a lovely experience also, in terms of a personal experience to be able to travel and to learn new methods that are done differently, in different labs. So even on a day-to-day basis, it was great. And yes, I certainly hope that our relationship will continue and might also generate future collaborations for sure.
So you mentioned Frédéric Ponvien as well, a collaboration with him. So did you meet him through the in-depth cost action as well?
I certainly did. Yes, absolutely. We met in Prague during the conference that INDEPTH organized in 2019, and I was able to extensively chat to him about my project. As I said, I personally consider him the most authoritative figure in this field about this topic, and we discussed at length, and I visited his lab in Perpignan during the summer of 2019 for more experiments. So it was certainly a fantastic experience as well, in terms of networking with other researchers and people with a lot of experience as well. Yeah,
fantastic. So I mean, I don't think I really need to ask this question, but you would therefore recommend STSMs? Yeah.
Absolutely, 100%. Like, it was really not just an enriching professional experience, but also very enriching personal experience. So I definitely recommend it. Yes.
So you didn't mention this, but as we're speaking now in November 2020, you've just had a paper in Plant Cell accepted, and I assume Frédéric, Aline, Christophe are all on that paper with you as well?
Yes, absolutely. So we're still doing some minor revisions, but we did hear that it has been accepted, and absolutely, yes, Aline and Fred are both collaborators of this first paper that we produced.
So it's fair to say that the techniques that you used in this plant cell paper wouldn't have been possible without the contribution of the cost action.
Absolutely. The experiments that were done in Clermont were done in Clermont because I wouldn't have had the opportunity to do them in my home institution, and so it was absolutely instrumental in the generation of really important data for this publication. Yes.
Excellent. Okay, well, thanks very much, Francesca, for joining us today, and thanks for your involvement in the cost action. It's been fun personally to get to know you, but it's great to see that your experiments have worked out so nicely.
Thank you so much, Geraint. It's been an absolute pleasure, and I believe that this work, it really is an in-depth baby, and it has really been pushed forward by the collaboration born through it. So thank you. Thanks a lot.